So hello everyone, I'm Debprosit, a second year MBA student from AIIMS Bhopal. So last time we were continuing with the chapter Principles of Inheritance and Variation, which was chapter number 4, and it dealt about Mendelian laws of inheritance and other laws of inheritance. So we were at 4.5 pleiotropy. We finished pleiotropy. So today we'll start from 4.6 sex determination. So, what is sex determination? The mechanism of sex determination have always been a puzzle for the genetics. Like how the sex is determined, we were not able to say it. So, what happened is there was a guy named Henking in 1891. He traced specific nuclear structures all through the spermatogenesis in a few insects. And it was also observed that 50% of the sperms received that structure and 50% did not. So he named the structure X body. And later we found out that the X body that Henking was talking about, it was in fact a chromosome. In fact, it was a sex chromosome, which was important for determination of sex of the organism. And it was given the name X chromosome. And it was observed that the Henking found, in insects, Henking's study was on insects, and in insects, the sex determination mechanism was XO type. And what do I mean by XO type is their eggs, the female gametes bear additional X chromosome besides the other chromosomes. So what happens in insects is, suppose there are 4 autosome pairs. So male gamete and female gamete. So male gamete will have 4 autosomal pairs plus 0 sex chromosomes, 0 0. And female gametes will have 4 autosomal pairs plus 1 extra X chromosome. And that's why this mechanism is known as exotype, where all eggs bear an additional X chromosome, and that determines if the gamete will be female or male. And sorry, this is not gamete, this is female. Sorry, there won't be O. Okay, because in gamete only one pair is coming. So the male gamete is 4 plus O type, and female gamete will be 4 plus X type. And here you can see that eggs fertilized by sperms having an X chromosome become female, and those fertilized by sperms that do not have an X chromosome become males. So what happens in insects is males form sperms. 50% of sperms have the X body and 50% does not have anything. And female produces egg. And now the sperms, if the X chromosome containing sperm fertilizes the egg, the resultant zygote will have all autosomes, nothing. And if the other 50% fertilizes the egg, then the genotype would be all autosomes plus 0 0. And they become males. They turn out to be males, and zygotes with this genotype turns out to be females. So this is the mechanism of sex determination in insects, and it is also known as XO type. And this X chromosome is known as sex chromosome because it was determining the sex of the organism, and the rest of the chromosomes were autosomes. They had no role in sex determination. So grasshopper is an example of XO type of sex determination, in which the males have only 1 X chromosome besides autosomes, and whereas females have a pair of X chromosomes. And So these observations led to the investigations of a number of species to understand the mechanism of sex determination. So what happened is now we started studying mammals, including humans, and we found that we do not have XO type sex determination. We have XY type of sex determination. So what happens in us is this is a human female and this is human male. So we have 23 pairs of autosomes. So let's suppose I write 46 plus XX is the genotype of female and 46 plus XY is the genotype of male. So what happens during gamete formation is 23 plus X will be one gamete and another gamete will be 23 plus X in case of females, and in males 23 plus X will be one gamete and 23 plus Y will be another gamete. So it's a chance factor. So females are producing 23 plus X type of gamete. 100% of gametes have X chromosome in females. I mean, eggs. And sperms, 50% of sperm has X chromosome and 50% of sperm has Y chromosome. So if sperm containing X chromosome fertilizes the egg, then the resultant will be 46 plus XX. And the zygote will turn out to be a girl or female. And if Y chromosome containing sperm fertilizes the egg, then the resultant would be 46 plus XY. And the child would be a male. And the Y chromosome is determining the sex of the child. So in mammals or in humans, the type of sex determination that we have is XY type. Okay, and it is known as male heterogamy. So what was happening in females? What was happening in insects was females had XX and male had XO. So it was also male heterogamy. And what is happening in humans or mammals is female had XX and male are having XY. So it is also male, which is heterogeneous. So this type of sex determination is called is known as male heterogamity. And there is also a female heterogamity which is seen in birds, and this system is ZW type. So what happens is female birds produce 50% egg with Z chromosome and with W chromosome, 100% sperms with Z chromosome. So if a sperm fertilizes the Z chromosome containing egg, the resultant will have ZZ. And if it fertilizes the egg containing W chromosome, the resultant will have ZW. So zygote would be female. And the female, it is known as female heterogamity, and it is ZW type of sex determination. And in sex determination in human, I already told how it happens. Male produces two types of gametes, one X and one Y containing, and female produces all of them. In female, all of them are X containing eggs. So if X sperm fertilizes the X egg, then the resultant would be a female, and if Y sperm fertilizes the X egg, then the resultant would be XY. So that is all what is explained in this part. So what have we learned yet is 3 types of sex determination. Starting with insect, exotype, then mammals, XY type, and then in bird, mammal or specifically, human, because birds are also mammal. And in birds, ZW type. These two were male heterogamety, and this was female heterogamety. And now sex determination in honeybee. So in honeybee, the mechanism is completely different. And it depends on the number of sets of chromosome. Does not depend on a single chromosome. So what happens is, okay, so what happens is in case of honeybees, females have a diploid cell. All of the cells in female's body will be diploid, and males are haploid. So every cell in a male body is haploid. They have only one set of chromosome. And in female, they have two sets of chromosomes. So a female undergoes meiosis to form gametes, and they form gamete. So one gamete contains haploid set of genome. So 16 chromosome in one gamete and 16 chromosome in another gamete. And male undergo gamete production by mitosis, and they just duplicate themselves, and the gamete will have 16 chromosomes. So now what happens is, if the egg is not fertilized, then it will remain haplontic, and if it remains haplontic, it will further develop into a male, and that's why males are haplontic in case of honeybees. And if egg is fertilized, if egg is fertilized, then the zygote would be diploid, and the diploids will further develop into female. Diploids are female, and out of female, who will be the queen and the worker bee depends on their diet. So here, from a union of a sperm and egg develops as a female, queen or worker. So queen and worker, all of them are female, and they all are diploid. They develop from fertilization, and unfertilized eggs develop as a male, which is or drone by means of parthenogenesis. So this is an important question. In which organism male is developed from parthenogenesis? The answer would be honeybee. And further, based on their diet, those who feed on royal jelly will turn out to be a queen, and those who feed normally, all of them will turn out to be a worker bee. So that is a different part. This is not that much important, but yeah, in honeybee, the sex determination depends on sets of chromosome. This thing is important, and this system is known as haploid diploid sex determination system. Males produce, and here males produce sperm by mitosis, and female produce sperm. Female produce egg by meiosis. And here, this is an important line which you can say, that males do not have father, and thus cannot have sons, but have grandfather and can have grandsons. So what does it means is, so here, this is a male. It undergoes mitosis, forms gamete, and the gamete, if fertilizes, then it produces a female. Okay, so males can have daughters, but because male will fertilize the female egg, and they will, and if it is fertilized, they will form female, but they cannot have sons. And male does not have father, because this is a male, and how it is forming is by female. Female undergoes meiosis, forms gamete, and this gamete is just converted into male. So there is no involvement of a male in forming of another male child. That's why you can see that this male child will have only mother, but not father. And similarly, male cannot also produce sons. They might produce daughters, but not sons. And that's why it is written that males cannot do not have father and cannot have sons, but have a grandfather and grandsons. So this female is formed by fertilization. So half of the genes are coming from female, and half of the genes are coming from male. So here's an involvement of male, and this is a generation before. So what do we call? It will be a grandfather. So this male child does not have father, but this male child have a grandfather, and this male will also be a and this male will also be a grandson. So they can have grandfather and grandsons, and this is a very important line. So now the next topic will be mutation. So what is mutation? Mutation is actually alteration of DNA sequences, and it results in changes in genotype, and it might also result in changes in phenotype. And mutation is another phenomena besides the meiotic combination, recombination, what happens during meiosis, that leads to variation in DNA. So what happens in mutation is it can be alteration in chromosomes, which is also known as chromosomal aberrations. Chromosomal aberrations, which are commonly observed in cancer cells. All of the cancer cells are formed by their own mutation. And mutation can also arise from change in single base pair of DNA. So this is a chromosome. It might go through some aberration. For example, suppose this part is deleted from the chromosome, so it will become like this, and now this is mutated chromosome, and here a large chunk of DNA is getting deleted. So this thing is known as chromosomal aberration. Now this is one method of mutation, and another method of mutation is this is a single strand of DNA, and for example, this is the sequence in that DNA, and this sequence gets changed. So for example, it becomes CG, and this is GC, and this is CG. Okay, so here you can see the sequence inside the DNA is getting changed, and this can also lead to a mutation, and this is known as point mutation. It happens in a point on the level of a single base pair, and the point mutation, the classic example of point mutation is sickle cell anemia, and deletions and insertions of base pairs causes frameshift mutations in DNA. So what is frameshift mutation is, for example, this is a DNA, and this was the sequence ACG, and this whole thing is a frame. Okay, now for example, this base pair gets deleted, and the new DNA would be like this: G, C, C, G, and there were multiple sequence like this, so the sequence will continue. But you can see that the whole frame, this whole frame of DNA is getting shifted by one base pair. It is getting shifted by every say base pair is getting shifted by one base pair, and this means the whole frame is shifting, and that is known as frameshift mutation. And the mutations can be caused by many chemical and physical factors, which are also known as mutagens. Okay, now coming to the genetic disorders. So what is genetic disorder? So first we will learn pedigree analysis. What is pedigree analysis? Pedigree analysis was based on heritability of a certain characteristic features in families. So why do we need pedigree analysis? Because as you can see in pea plants, we were just crossing random plants based on our will, and we were obtaining the results. But in human, this is not possible. We cannot say two humans to say that you two fertilize together and we want to study your child for scientific purposes. This is not possible and this is not ethical. So what happens in human, we study their family line. We go backwards from in family relations, and this thing is known as pedigree analysis. We study the family line. So study of family history about inheritance of a particular trait provides an alternative. So this is an alternative approach to study the inheritance of certain traits, and such analysis of traits in several generations of family is called pedigree analysis. And pedigree analysis, in pedigree analysis, we study the inheritance of a particular trait and it is represented in the form of family tree over generations. And okay, that's it. And that's how and by studying the family tree or family history, we can trace the inheritance of a specific trait, abnormality, or disease. Okay. Now and these are some of the notations that we use in pedigree analysis. So square box means male, round means female, and this diamond shape means sex unspecified. And if it is filled with some color, then it means that affected individuals, and this thing shows mating, and this thing shows consanguineous mating. It means mating between relatives, or most commonly cousins, and this means their parents, and these are their children, and this means these are parents and they are producing a child which is having its box filled with solid color. So this child is affected, and this thing means, I mean, this you can confuse it with this sex unspecified. So sex unspecified is just a diamond shape, and if someone writes a diamond shape and writes a number in between it, like for example 6, so it represents 6 unaffected offsprings, and this shows 5 unaffected offsprings. But this is not used in general purposes, so you won't probably see it being used in questions. So Mendelian disorders. So what is Mendelian disorders? So genetic disorders can be classified into 2 groups. One is Mendelian disorders and another one is chromosomal disorders. So Mendelian disorders are determined by mutations in single gene, or the mutations are at small level. Only a single base pair is getting mutated, or there is having some sort of change. So the entire the structure of entire chromosome is remaining unaffected, and that's why remaining unaffected, and it and the affected gene or the mutated gene follows the laws of inheritance that were provided by Mendel, and that's why these disorders are known as Mendelian disorders. And in chromosomal disorders, you can see massive chromosomal aberrations. Like, for example, suppose this part of chromosome, it will get deleted, completely deleted. That's it. And the new chromosome will be like this. This is chromosomal aberration. And some examples of Mendelian disorders are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. All of these are those in which only one base pair is getting affected, one base pair is getting mutated. And here you can see pedigree analysis of a family. So this is representing autosomal dominant disorder, and this is representing autosomal recessive disorder. So how do we find autosomal recessive disorder? There's a very simple technique. So just look for unaffected parents producing an affected child. If this thing is anywhere, if you see this thing anywhere in your pedigree analysis, it will be a recessive disease. And it is an autosomal recessive disease because it is happening in both female and male. Okay, so here we can see that unaffected parents producing affected child, also unaffected parents producing affected children, and this thing says that it is autosomal recessive. Now you can understand it by I can explain it by using genetics also, I mean by using genotype also. For example, suppose this the recessive gene is A, and this is the disease causing gene, which means when the recessive gene is present in homologous condition, the disease will be expressed. Otherwise, on being present with a dominant gene, the disease will not get expressed according to the law of dominance that was given by Mendel. So for this child to be smaller, their parents must be capital S small and capital S small. See, these parents are not affected and are an affected child. So both of them must be heterogeneous in order to produce this. And that's the concept. And here it is an autosomal dominant representation of myotonic dystrophy. So you can explain, you can just understand it by this, that autosomal gene, this is the mutated gene, and this is causing disease. So disease free genotype would be small, and rest other capital small and capital, both of them will be diseased. So this one is small, we can say it for sure because it is unaffected in case of autosomal dominant disorder. And this parent, she will be capital A small a because she is diseased. But look at this, among their children, not all of them are affected. So that's why she can't be capital A, because if she was homologous dominant, then all of their child would have had capital A small a type of genotype, and all of them would have been affected, but not all of them are affected. That's why we can say that this female parent is having an heterogeneous or heterologous chromosomal arrangement or heterologous genotype, and this male parent is having homologous recessive genotype, and their children are having all of the unaffected ones are having recessive homologous genotype, and all of the affected ones are having heterologous genotype or heterogeneous genotype. Okay. Now, the example of some diseases will discuss some of the diseases. So, starting with color blindness. Color blindness is a sex-linked recessive disorder, and the genes. I mean, there are certain genes in X chromosome which are mutated, and these mutated genes are recessive. So when these recessive genes are transferred to the next generation, the color blindness is produced, and it occurs in about 8% of males and 0.4% of females. And let us draw a diagram to explain it in a better way. So this is X chromosome, this is X chromosome, this is female, and this is XY of male. Okay. Now, for example, suppose X dash, this represents the mutated gene. So the mother is carrier and the father is. So what happens in during fertilization fertilization is there are 4 possibilities. One is X dash X, another one is X-Y, another one is X-SX, and another one is another one is X-X. Wait a minute. One is X-X, another one is X-Y, and the next one will be just X and X, and another possibility will be XY. So these are 4 possibilities. So you can see among the female child, one will be carrier and she is unaffected. So for female to be affected, we need both of the X chromosomes to be mutated. Both of them should be X-X type. Unless this is happening, a female will never be colorblind. So for female to be colorblind, we need the father to be colorblind as well, because father should have X dash. If father is having normal X chromosome, female child will always have a normal X chromosome and she will never develop color blindness. But in case of male, as you can see, only one mutated X chromosome is sufficient to produce color blindness. So if the mother is a carrier, then 50% of the male child would be colorblind and 50% of the male child would be unaffected. And this is what is written here, that son of a woman who carries the gene, or means who is a carrier, has 50% chance of being colorblind. And that's what I expressed. I mean, that's what I that's what I drew here in form of a diagram. So the mother is not herself colorblind because the gene is recessive and the mother is carrier. It means mother is having heterologous genotype, and that means that its effect is suppressed by her matching dominant normal gene. So daughter will not normally be colorblind unless her mother is a carrier and her father is a colorblind. So this is what I said, that for a female to be colorblind, both of the parents should have at least one affected gene, at least one affected X chromosome. So that means that the mother must be a carrier and the father must be a colorblind. Now coming to hemophilia, the concept is similar to color blindness. This is also a sex-linked recessive disease, and so the inheritance will be similar to colorblind. So I'm not going to explain it here. So what happens in this, rather we'll see the phenotypic aspect. So what happens in this disease, a single protein that is part of a cascade proteins involved in the clotting of blood. So as we know that clotting of blood is a cascade process, several different reactions are linked together. So in these reactions, one single protein, which is factor 8, this single protein is mutated and the entire process is disrupted, and there is non-stop bleeding in case of any injury. The clotting does not occur rapidly, and this is what happens in hemophilia. And the possibility of a female becoming hemophilic is extremely rare because mother of such female has to be a carrier and the father has to be hemophilic. And hemophilic individuals individuals who are having the expressed phenotypes of hemophilia, they do not live to the later stages of their life. They generally die earlier by excessive bleeding and all. So this possibility is very, very rare. So we do not normally see hemophilic female, but hemophilic male is rather a more possible occurrence. And the family pedigree of Queen Victoria shows a number of hemophilic descendants, and that's how we discovered the disease hemophilia also. So this is a part of history. You can search it on Google and read more about it, how the study of Queen Victoria family pedigree helped us in discovering the disease of hemophilia. Now coming to the next disease, sickle cell anemia. So what happens in sickle cell anemia? It is an autosomal linked recessive disease. So hemophilia and color blindness were X-linked recessive, and here sickle cell anemia is autosome linked recessive disease. So what happens here is this disease is controlled by a single pair of allele HPA and HPS. So out of the three possible genotypes, only homologous individuals, means those containing HPS and HPS, will have the disease, and heterozygous individuals, they will remain they will apparently they will appear apparently unaffected, but they are carrier of the disease. HbA and HbS1 are carriers, and HbA and HbA genotypes with this genotype, individuals with this genotype are completely unaffected. So this is the main mechanism of sickle cell anemia or inheritance of sickle cell anemia. What happens here is you can see that this gene, this is a normal HbA gene, but in sickle cell anemia there is a mutation in this part. See, this is a point mutation in a single base pair, and this is a sickle cell gene, HbS gene. So the mRNA produced by this gene will be having a GAG codon. The codon is GAG, but the mRNA produced by sickle cell gene is having a codon GUG, and this is HbA peptide and this is HbS peptide. So in HbA peptide, at the 6th position, the amino acid is glutamine. But in the HbS peptide, at the 6th position, the amino acid is valine. All other amino acids are just like that. Only the amino acid at the 6th position is changed, and that is causing this major phenotypic change in RBC structure. So what happens is individuals who are having sickle cell anemia, they will have HbS peptide in their RBC, and HbS peptide, what happens when there is a condition of hypoxia? In case of hypoxia, these HbS peptide undergo polymerization, and when they undergo polymerization, the RBC becomes sickle shaped like this. Normally the RBC is this, but when there is hypoxic condition, these peptides will undergo polymerization, and the RBC will be like this, and it can create clots in blood vessel. It does not pass through the blood vessel efficiently, and it can clot the blood vessels and can cause several what should I say complexities. Now coming to phenylketonuria. So what is phenylketonuria? It is an inborn error of metabolism. So there's an enzyme that converts phenylalanine into tyrosine. This is phenylalanine hydrolase. So in these individuals, the gene that is producing PA hydrolase enzyme, it is mutated, and the PA hydrolase enzyme is absent. So as a result, the phenylalanine gets accumulated and it is converted into phenylpyruvic acid, and phenylpyruvic acid further accumulates in brain, and it can cause mental retardation, and it can also be excreted through urine, and because it is absorbed poorly by kidney. Okay, so this is PKU or phenylketonuria. And coming to the next disease, thalassemia. This is another autosome linked recessive disease. So what happens here is when both the partners are unaffected carriers for the gene or heterozygous, the defect could be either due to this is transmitted when both of the parents are carrier. Okay, now it can happen either due to reduced rate of synthesis of one of the globin chains that make up the hemoglobin, either due to mutation or deletion So, which ultimately results in the reduced rate of synthesis of one of the globin chain, alpha and beta. These two are globin chains. So, this is the structure of an RBC. This is Fe plus 2, Fe plus 2, Fe plus 2, and another Fe plus 2. Two of the polypeptide chains are alpha and two of the chains are beta. Okay. But in thalassemia, what happens is either one of the polypeptide chains may have some deformed, may get deformed. So either alpha can get deformed or either beta can get deformed. So if alpha is deforming, then it is known as alpha thalassemia. And if this beta is deforming, it's known as beta thalassemia. So in alpha thalassemia, what happens is production of alpha globin chain is affected. And in beta thalassemia, production of beta globin chain is affected. And how it is maintained, how it happens is, in case of alpha thalassemia, there's a chromosome, 16 chromosome. And in 16 chromosome, we are having two genes, HPA1 and HPA2. These two genes are responsible for production of alpha globin chain. And when there's a mutation of either of the gene or both of the genes, the alpha globin chain gets affected and the alpha globin chain becomes deformed. And the thalassemia that is produced is known as alpha thalassemia. In case of beta thalassemia, 11th chromosome have only one gene, HBB. So if there is any mutation of this HBB gene in the chromosome number 11, then the beta globin chains get deformed and it is known as beta thalassemia. And thalassemia differs from sickle cell anemia in that thalassemia is a quantitative problem of synthesis of too few globin molecules, while sickle cell anemia is a qualitative problem. So what does it means that in thalassemia, if only one gene is getting affected, one gene is getting mutated, the effect of thalassemia will be less potent than when both of the genes are getting mutated. When both of the genes are getting mutated, the potency of thalassemia, the expression of the disease will be more. So it depends on a quantity. I mean, it depends on the quantity of mutation. So that's why it is a quantitative problem. But in sickle cell anemia, it is just, if it is mutated, the disease will be expressed. There is no possibility of quantification in this disease. There is nothing such that कि एक जीन का म्यूटेशन थोड़ा ज्यादा हो रहा है या फिर एक जीन का म्यूटेशन थोड़ा कम हो रहा है. इट इज कि एक जीन का आइदर म्यूटेशन हो रहा है और म्यूटेशन नहीं हो रहा है. एंड इफ म्यूटेशन इज हैपनिंग देन इट इज सिकल सेल एनीमिया, एंड इफ म्यूटेशन इज नॉट हैपनिंग देन इट इज नॉट सिकल सेल एनीमिया. And that's why sickle cell anemia is a qualitative problem, and this is a very important point. And there will be several questions in NEET from this point. So these were the Mendelian disorders. So we were observing mutation at the gene, at the single base pair level, at the genetic level in Mendelian disorders. And in case of chromosomal disorders, what happens? Chromosomal disorders, there can be aneuploidy and there can be polyploidy. So what happens is, in case of chromosomal disorders, for example, these two are homologous chromosomes, and during the metaphase 1, so generally what is supposed to happen, these two chromosomes are supposed to get segregated from each other. So segregation should happen. But if in metaphase 1 segregation process, sometimes the entire pair of sister chromatids comes to one pole. I mean, two chromosomes are here, and this side has no chromosome, and the cell divides, and finally, in the daughter cells, in one cell will be having one set less, and in another cell will be having one set more. So failure of segregation of chromatids during cell division results in gain or loss of chromosome. This is known as aneuploidy. Okay. This is aneuploidy. And polyploidy is failure of cytokinesis after telophase stage of cell division results in increase in the whole set of chromosomes. That is known as polyploidy. So aneuploidy is when only one or two chromosomes, I mean, the entire set is of 23 pairs. So when there is increment or decrement of one pair or two pair or maybe even three pair, this thing is known as aneuploidy. But when the entire sets is getting increased, I mean, if the daughter cell is having one daughter cell is having 46 pair, another daughter cell having 0 pair, this condition where the entire set is just getting doubled, this is known as polyploidy, and this is known as aneuploidy. And the mechanism of both aneuploidy and polyploidy is different, and it is explained here. And so the chromosomal, so in chromosomal abnormalities, in chromosomal disorders, what happens? There is a problem in either metaphase 1 or telophase 2. So it is a problem of meiosis. The meiosis process is having problem, and that's why the disease is getting expressed. And there are several diseases. So first we should start with let's start with Down syndrome. So in Down syndrome, what happens is trisomy of 21st chromosome. So there's an additional copy of the chromosome number 21. So in an individual, this is the karyotype of an individual. So you can see that chromosome number 1 has one from mother, one from father, one from mother, one from father, one from mother, one from father. So all of the chromosomes are normal and they are 2 in number. They are 2 in number. Okay. But what happens is, see, 21st pair, here we're having 3. Normally there should be only 1 pair, or there should be only 2 21 chromosomes in an individual. But in an individual of Down, in a patient of Down syndrome, we see that there are 3 21 chromosomes. And how it happens is, one is from father and other 2 are from mother. So while forming the egg in mother, the 21 sister chromatids, they did not get separated from each other, and when the final gamete formed, final gamete was having 221. So when this sperm having only 121 fertilized this egg, the zygote now have in total of 321 chromosomes. It can also be vice versa, where the father is producing the sperm having 221 chromosomes, and mother is producing egg having only 121 chromosome. So this is Down syndrome, and it was discovered by Langdon Down in 1866. And the examples, I mean, the features of this individual will have the individuals will have short stature with a small round head, furrowed tongue, palm is broad, physical and psychomotor and mental development is retarded, and this is the diagram. And in Klinefelter syndrome, something similar to this happens. I mean, this is a genetic disorder caused by non-disjunction of chromosome, and the individual is having XXY character, XXY type of chromosomal arrangement, and so it is having Y chromosome. So that means they're actually male, but they have 2 X chromosome. So there will be some phenotypic changes like gynecomastia, development of the breast, which is also known as gynecomastia, is expressed, and they will also be sterile. So this is the example of Klinefelter syndrome, and the genotype is XXY. And Turner syndrome. In Turner syndrome, what happens is, in such disorders, there is absence of 1 X chromosome. So in females, we generally have two X chromosomes, but when there is absence of an X chromosome, the genotype will be XO-type, and such females will be sterile, and ovaries will be rudimentary, and other features will, including secondary sexual characters, will also be rudimentary, and the patients are known as, this disease is known as Turner syndrome. So these were some chromosomal disorders, and there is the summary. So we finished the entire chapter. So what happened is we started with Mendelian genetics, and Mendel proposed laws based on his experiments on pea plants, and we saw some exceptions of Mendelian genetics also here, in form of incomplete dominance and codominance, and also the concept of inheritance of two genes, how a dihybrid cross happens, all the Punnett square, and how the law of independent assortment was given. So these were Mendelian laws, and we also came to know about the chromosomal theory, which was given by Sutton and Boveri for inheritance, and this box, which is particularly important. And this is chromosomal inheritance. And after that, based on the experiments of T.H. Morgan and Drosophila melanogaster, the sex-linked or the concept of linkage or recombination. So we learned about how linkage happens, what is a combination, and things like this. And after that, polygenic inheritance, which was basically human skin color or human height, how different, how multiple genes are affecting a simple A was just one character, and in pleiotropy, one gene was having multiple characters. So these two were something of contrasting to each other. And then we learned about sex determination in different insects, humans, and birds, and also in bees, honeybee. And after that, we went to mutation, then genetic disorders. We learned how to do pedigree analysis. We learned about Mendelian disorders. And after that, we learned about chromosomal disorders. And that's how the chapter ended. So let us solve the question answers of this. So let us solve the question answers. Okay. So first question is, mention the advantages of selecting pea plants for experiment by Mendel. So I explained it already, and as you can see in this answer, I have written the answer, because they show variable contrasting characters, they're bisexual, so we can produce breeding varieties, and all of the features. It is given very well in NCERT, and I have already discussed it, so this is done. Now differentiate between the following: dominance and recessive, homozygous and heterozygous, monohybrid and dihybrid. So all of this were explained in the chapter. So you can read NCERT, and I have written it here too. As you can see, dominance expresses itself irrespective of the presence of recessive allele, and in case of recessive, this does not happen. And also in homozygous means two similar allele for a trait, and heterozygous means two different alleles for a trait. And in monohybrid, as you can see, monohybrid means that only one contrasting character is taken while. In dihybrid, two contrasting characters are taken while crossing. And a diploid organism is heterozygous for 4 loci. So how many types of gametes will be produced? So this is the example. Diploid organism heterozygous for 4 loci. So this is just permutation combination. You can just write all the possibilities. Here I found the possibility of forming 8 gametes, so the answer will be 8. Now explain the law of dominance using a monohybrid cross. So this was so now the law of dominance. Explain the law of dominance using a monohybrid cross. So this was well explained in the chapter also, and I have explained it here too. That you take monohybrid characters and make a Punnett square. So and I have already shown how to make a Punnett square. So this will be the answer. Now define and design a test cross. Wait a minute. So define and so this is complete. And define and design a test cross. So we have already read the definition of a test cross. We use test cross to experiment the genotype of an organism. This is generally a cross between a dominant phenotype and a recessive phenotype. And this is the phenotypic diagram. I mean, this is the genotypic diagram of the test process. Okay, so this is the design of a test process. Now, using a Punnett square, work out the distribution of phenotypic features in the first filial generation after cross between homozygous female and heterozygous male for single locus. So this is it. Simple. They have. This is actually the question of a simple test cross, and they have asked us to make a Punnett square. So I made a Punnett square here, and you can see the ratio was calculated and everything of this. I mean, I have already discussed all of this in the chapter itself. Now, question number 7. Make a cross between these two genotypes and write the proportion of phenotypes of tall and green and dwarf and green. So this is the cross, as you can see, and we can divide it into two monozygous cross. So I have divided it into two monozygous cross, and and see the phenotypic ratio of tall and dwarf is 3:1, and the phenotypic ratio of yellow green is 1:1. Now, when we are combining both the characters, that the occurrence of tall and green both together, then we have to multiply the possibilities of both of these. So the possibility of tall plant is 3 out of 4. Possibility of green plant here is 1 out of 2. So upon multiplying, we're getting 3 out of 8. So the answer would be 3 out of 8 for tall and green, and similarly in dwarf and green. So possibility of dwarf plant is 1 out of 4. Of a yellow plant is 1 out of 2. So 1/4 multiplied by 1/2, the answer would be 1/8. And this was already discussed by me in the chapter. It's chapter 2. And now coming to the next question. Two heterozygous parents are crossed. If the two foci are linked, what will the distribution of phenotypic features in F1 generation for a dihybrid cross? So if the genes are linked, they behave as a single gene. And we have also read that in the chapter while discussing linkage, that if two genes are present on a single chromosome, they're linked to each other and there is no recombination, then both of the genes will behave as a single gene. And the phenotypic ratio will be 3:1, and that is it. So whenever you see linkage is described, I mean, linkage is written in the question, and there are 2, 3, 4 genes, but the question have specified that all the genes are linked. What will and are asking what will be the and there is no recombination that is written. So, and then you should consider all of the genes together as a single gene, and the phenotypic ratio will always be 3:1. This will always act as a monohybrid cross. When there are so the concept is when there is linkage and no recombination, the cross will behave as a monohybrid cross, no matter how many genes are involved. Now, question number 9. Briefly mention the contribution of T.H. Morgan in genetics. So it is already mentioned in NCERT 2, and I have written the points here that he used fruit fly. He gave the theory of linkage, and he proved that tightly linked genes shows less combination and recombination, and loosely linked genes showed higher recombination. Okay. Now, chromosome number 10. What is pedigree analysis? So I have discussed it today. What is pedigree analysis, and suggest how such an analysis can be useful. So pedigree analysis is the study of family history, and it helps in knowing the inheritance of a trait. It is further, I mean, it is very much important in case of humans, because in humans we cannot just perform the desired cross. We cannot just say to humans to cross among themselves, and we cannot just take their child in part of our study. So in case of humans, we take family history, and it can be used to know the characteristic of future generations too. We can just, we can trace the inheritance of a mutated gene or of an affected character. This is the significance of pedigree analysis. And how is sex determined in human beings? So I have already discussed it, that in human beings, sex determined by Y chromosome. Half of the sperms are having Y chromosome and half of the sperms are having X chromosome, and that's why if a sperm containing Y chromosome fertilizes the egg, the product will be a male, and if a sperm containing X chromosome is fertilizing the egg, the product will be female. I should not say product; it will be the offspring. So the offspring will be male or female. Okay. And question number 12, that a child has blood group O and a father has blood group A and mother has blood group B, then what will the genotype of parents? So child is having a blood group O. Okay. Child is having blood group O. It means child is having genes IO and IO. And father is having blood group A. Mother is having blood group B. For having O genes, would have at O gene, and father must have 1 and 2, and mother is IAIO, mother's is IBIO, and child is having genotype IOIO. So these are some simple questions that you can expect from this chapter. Now explain the following terms with example: codominance and incomplete dominance. So we have explained it in detail already. Codominance is when both the characters are present simultaneously, and incomplete dominance is when none of the character is present, but the dominant character is slightly repressed in the offspring. This is known as incomplete dominance. I mean, the dominance is not completely expressed. And example would be codominance is blood group IB. Blood group AB blood group. And example of incomplete dominance is flower color in Mirabilis jalapa or snapdragon. And question number 14, what is point mutation? Give one example. So I have shown point mutation. I have described point mutation today. In case of sickle cell anemia, point mutation is when a single base pair is mutated, and the classic example is sickle cell anemia, where we are having a point mutation of a base pair to U. So the GAG codon is getting changed into GUG codon, and at the 6th position, the GAG was producing glutamic acid initially in the unaffected individual, but now in the affected individual, GUG codon is producing valine, and this is the example of point mutation. And question number 15, who had proposed the chromosomal theory of inheritance? So the answer is Sutton and Boveri. And question number 16 mentioned any 2 autosomal genetic disorder with their symptoms. So we have read it today only, that one was phenylketonuria and another will be sickle cell anemia. And you know that in phenylketonuria, PA hydrolase enzyme is getting mutated, and that's why the phenylalanine is getting deposited, and it is converted into phenyl pyruvic acid. And phenyl pyruvic acid, when deposited in the brain, it is causing mental retardation, and it is also getting excreted through urine. And in sickle cell anemia, what is happening that RBC is getting affected, and RBCs are becoming sickle shaped when there is presence of HBS gene. The reason is the RBCs containing the individuals containing HBS gene are producing the polypeptide, producing a different polypeptide which undergoes polymerization when there is hypoxic condition, and that's why the RBCs convert into sickle shaped, and this sickle shaped RBCs can cause several complications by accumulating in blood vessels. And this is it. All of the questions are discussed by me, and the entire chapter was discussed by me, and this was a brief discussion, so you have to read this chapter in detail, and also take help of external resources too, where you do not understand the concept very well, because in NCERT everything is written in very precise way. So in some part you might have to refer to external resources, or you can just search it on Google to have some more information too. But ultimately, restrict your preparation to NCERT and what NCERT what is given in NCERT, and try to read it again and again and again. The more number of revisions you do, the better grip of that chapter you will have. And that's it. Bye.